Good afternoon, ladies and gentlemen, and welcome to the New Developments in GRADE on behalf of the GIN webinar series, conference call hosted by Dr. Holger Schunemann. My name is Julie, and I'm your event manager. During the presentation, your lines will remain on listen only. If you require assistance at any time, please key star and then 0 on your telephone, and a coordinator will be happy to assist you. You may submit web questions throughout the presentation by clicking on the Q&A tab indicated by a question mark. Type your questions and send to all panelists. These will be addressed in the Q&A session. You can also ask an audio question, and to do this, please key star and then 1 on your telephone, record your name, and press the hash key. I would like to advise all parties this conference is being recorded for replay purposes, and now I'd like to hand the call over to Dr. Holger Schunemann. Please go ahead, sir.
Thank you very much. Welcome, everyone. This is the second webinar in a relatively short time that I will be giving. My name is Holger Schunemann. I work at McMaster University. I'm a guideline methodologist, guideline developer, and practicing internist. Today, I really represent a number of organizations, but I'm giving this talk on behalf of the Guideline International Board of Trustees, of which I am a member, and I will move on to my disclosures. I'm co-chair of the GRADE working group. Today's topic obviously is GRADE. I've worked with a number of organizations, and I sit on the steering group of Cochrane, and as I said, the board of directors or board of trustees of GIN. I don't have any direct financial conflicts of interest today, and there are a number of colleagues from the GRADE working group that provided material that I will be using. So just briefly, as a background, it's always good to know where am I speaking from, or where are we housed. So this is obviously a map of the world. We are McMaster based in North America, specifically very close to Toronto in Hamilton, just between Toronto and Buffalo on the way. Our department was founded by Dave Sackett. It's widely considered the birthplace of evidence-based medicine. And McMaster is the home of problem-based learning developed in the 60s and 70s. If you want to know more about us, you can just Google us about our wonderful department. There are about 40 full-time professors who work on topics that are relevant for what we will be speaking about. So today's presentation really is an update from the GRADE working group. And you've heard the moderator, you can submit questions both verbally by pressing the buttons appropriately on your phone or via the chat, and I will try to make sure that I see those questions. So I'm turning on my chat, but once again, through the moderator, you can definitely get a hold of me and ask these questions. That reminds me of the presentation style. So it is structured in a way that there will be about 45 minutes of presentation. There should be sufficient time for questions. There are also some questions that I have for you. I will not ask for answers, but they are kind of a self-checking, a self-test. And you see that I have a very long list of topics that I would like to provide an update on. We may not get through everything depending on how many questions you ask, and we feel that the interaction is more important than necessarily taking you through everything that I have on this list. So if there's a lot of interaction through the chat and these questions or comments are considered critical for moving on, then we may just stop a little bit early, and that's very possible today given that we're dealing with an update. So I will clarify a little bit of terminology about GRADE and have a very brief intro for those of you who haven't heard much about GRADE before. Talk as in the announcement about GRADE software, the GRADE profile or GRADEpro in short. Talk about the use of GRADE in tests and focus there on the presentation of information, GRADE in non-randomized studies. And I mentioned already here that some of the work that I will be presenting in this context will require further work. Then I will speak about the GRADE project groups, provide some background there. So for instance, GRADE for animal studies and rapid evidence assessment, which is under development, and then GRADE guidance on good practice statements. And this will not happen necessarily in this order, close to in this order, but it will flow better if some of these items are being touched on repeatedly, and with when they belong, in particular if they belong to other sections that I will be covering. Okay, so GRADE, just as a brief background, has developed a unifying, transparent, and sensible system for grading the quality of evidence, more on that in a little bit, and developing recommendations. Most of you will know that over 90 organizations adopted or use GRADE, including some very major organizations that are mentioned here, the Cochrane Collaboration, the World Health Organization, the National Institute of Health and Care Excellence in the UK, and so on. And the GRADE really, as a brief background, generally deals with assessing the certainty in evidence, and that includes involving the assessment of evidence in a transparent way. It has to do with assessing the confidence in an estimate of an effect, and that starts with a single study and ends with a body of evidence by outcome, and leads to typically, often, to a recommendation, but not all organizations will actually develop recommendations. So, for instance, Cochrane uses GRADE to assess the certainty in the evidence in a systematic review. Developing structured healthcare recommendations, according to GRADE, also involves making judgments and decisions transparent. My last webinar focused on the evidence to decision frameworks, which will be a very brief topic today. It is based on a comprehensive list of criteria that influence a recommendation, and ends with clearly developed and formulated messages for action, and that should facilitate adoption, adaptation, or de novo creation of recommendations. So how does the GRADE working group work? I included this here as an update because we do get a lot of questions, and it might be helpful to disseminate via webinar. Membership in GRADE is free. GRADE actually is an international collaboration working group. We hold 2 or 3 annual meetings, and subscription to those is really on a first-come basis. You can sign up for membership on GRADE's website that you can Google, and that I will show in a second. And as I said, it's an informal international group with, however, formal networks and centers around the world. So the first GRADE center was indeed at McMaster University. The last edition is the UK GRADE network, and here you see a screenshot with the current approved GRADE centers and GRADE networks around the world that you can relate to. The main goals of the working group are to advance the GRADE approach, obviously to conduct methods work in the certainty of evidence assessment and recommendation development. We collaborate with many organizations, so we have a formal collaboration with GIN and Cochrane, and we support WHO as well as other organizations with GRADE methods. So I'll start with some clarification about terminology. So GRADE now uses the term certainty in the evidence in order to make sure that we are usable, or GRADE is usable by different organizations that prefer different terminology. So GRADE now uses certainty in the evidence synonymously to quality of evidence and the confidence in the effect estimate. Our preferred term is certainty in the evidence, and for most of the rest of the presentation, or largely for the rest of the presentation, I will refer to the quality or confidence in the effect estimates as certainty in the evidence. A few other issues. So the GRADE approach or GRADE system really describes GRADE's conceptual underpinnings, what I basically just described on the previous, one of the previous slides. So certainty of the evidence and developing structured healthcare recommendations, supporting decisions. The GRADE criteria relate to the evidence to decision frameworks. Those are the decision criteria in the assessment of the evidence. So in the evidence to decision framework, the criteria are supported or determined by our decisions are made based on the about the criteria based on judgments and research evidence and additional considerations. And then there are grade domains within these criteria. So, for instance, in the certainty in the evidence domain criterion, there are 8 domains to assess the certainty in the evidence. I will get back to that briefly, but most of you will be familiar with that as well. And then finally, within the domains, there may be items to assess, such as for inconsistency, we would be looking at overlapping confidence intervals, I-square, and p-value to assess that. So some clarification about that terminology for the rest of the presentation. Moving out of the grade criteria, but we are often asked, what are really the minimal criteria for saying that you or your organization are using grade? And that was recently updated by the grade working group. So there are 6 criteria. One is the definition of the certainty of the evidence should be consistent with grade writing, and there are many publications, obviously, that describe that. Each of the grade domains for assessing the certainty in the evidence should be considered. It is fine if you use different terminology. So if you prefer using heterogeneity for inconsistency or limitations in the detailed study design and execution instead of risk of bias, that is fine. The certainty in the evidence that is assessed or should be assessed for each important outcome that determines a recommendation. The 4th criterion is that the evidence summaries and evidence to decision criteria should be used as a basis for judgment about recommendations, and that ideally should be based on systematic reviews. At a minimum, the evidence and the methods used to summarize the evidence should be clearly described. And when moving to recommendations, an explicit consideration of each of the grade criteria either should be done or it should be explained what approach and which criteria are being used. So if you look at recommendations from an individual patient perspective, your approach to using costs in the evidence to decision framework may be different. So ideally, grade evidence to decision frameworks should be used. And then finally, the strength of recommendations should be assessed using 2 categories, either for or against an option, with grade definitions such as strong recommendations and weak recommendations. Alternative terms also here are perfectly okay. So using conditional or discretionary recommendations, that we look at that only as an alternative term. Conceptually, this is not different for the grade working group. So very much related to this, an update relates to grade and environmental health. This was mentioned in the announcement of the webinar. This is from a paper that was recently published. Oftentimes, questions have been raised if grade can be used based on the criteria that I just described for rapid assessment of evidence. So this paper, very important, is not yet official grade guidance, but a grade project group is beginning to work on this. And I will just quickly take you through this. The reason for providing information on how to use grade in the context of urgent or emergent situations, fulfilling the grade criteria, is that organizations, for instance, in environmental health, but also in other areas, are looking for structured frameworks for evidence synthesis and assessing the certainty in the evidence. So they need to be fit for purpose. And sometimes systematic reviews are not possible to assemble the evidence. For instance, if you need to provide an answer in an emergency response. We believe that grade certainty in the evidence domains are still helpful. This is a table from that paper that I just described that is available on PubMed. In that paper, we begin by categorizing the type of response in terms of the certainty of evidence assessment. So, and these categories could include very or ultra-short emergency responses that need to be given within an hour or very short, within a very short period of time. So, for instance, when there are toxic spills in the environment, an example is provided here. There are urgent responses that may take 1 or a couple of weeks, or where the time frame is 1 or 2 weeks, and we believe that grade can still be used under those circumstances. Ideally, there's an available direct systematic review or at least indirect systematic review that deals with a similar intervention or exposure Then there are rapid responses that may take 1 to 3 months. The rapid advice guidelines by WHO and avian influenza from about 10 years ago are a good example. And then obviously the routine responses. And this table, for those of you who are interested, summarize how the GRADE domains could be used, the various GRADE domains that we are very familiar with, an overall judgment about the certainty in the evidence for an outcome, and a possible summary statement. Okay, I'm now coming to a self-assessment. Once again, this is also a reminder or an encouragement for letting me know if you do have any questions through the actual chat. So the self-assessment is now, the question is, GRADE requires systematic reviews. The answers are A, yes, GRADE cannot be used without systematic reviews. B, ideally yes. C, if systematic reviews are not used, the methods of compiling and rating the evidence must be transparently described. Fourth option is B and C. I'll give you 1 second to check your understanding of what I have presented so far to possibly come up with an answer. And once again, encouragement to actually do use the chat. And our coordinator on the phone will advise me as you have questions or issues that are being raised. And the answer to this question is B and C. So ideally we use systematic reviews, but if systematic reviews are not used, such as in an emergent situation, it is helpful to describe how the evidence that was used has been actually used, and working through a structured process may still be helpful. Okay. I move on to the next update, update on the GRADE application or GRADEpro. So now that becomes very practical for guideline developers, those of you who are active guideline developers. So GRADEpro.org is our website. This is the official web software of the GRADE working group. You can log in through that website, and the main purposes and current content of GRADEpro are the following. It really assures that GRADE is used correctly. It allows you to produce evidence profile and summary of findings tables, use the evidence to decision frameworks in an interactive format. It includes the GRADE handbook. It provides recommendations for how to use GRADE for practice. As I said, it has interactive tools that help with implementation as well as research possibly in your setting, provides decision support, and really guides you through the process of guideline or recommendation development. There are over 12,500 users now, over 3,000 panel members have been involved, and there are now 30,000 questions created, with about 1,000 evidence to decision frameworks, of which 500 have recommendations. It is free for non-for-profit use in its basic function, and the if there ever is any licensing, it is being used for maintaining and reprogramming GRADEpro. I just show this slide. This will be made available. You can go back to that. There are a number of avenues to contact individuals who work on GRADEpro, but really your feedback will be used to further improve this tool. So there are different entry points into GRADEpro depending on what the target or what the intended use is. You can choose to just produce an evidence summary or a full guideline, or do an evidence to decision framework. You can manage conflicts of interest according to different guidance from different organizations. For individuals involved in your guideline panel, this essentially is the list of issues or tasks that can be completed with GRADEpro. I'm showing you one example here. The GRADEpro now allows question formulation in more detail and outcome selection. So in your PICO framework, selecting outcomes that are important for decision making, with brainstorming, rating the importance, and a final list of approval of the outcomes, and all of that can be done online. It obviously includes the development of evidence profiles and summary of findings tables. And finally, and this is really new, you will have to request administration rights for governing the evidence to decision frameworks that you can use now online. So while we use the evidence-to-decision frameworks in actual panel meetings by projecting on screen, this can also be done online through, for instance, voting processes on individual grade criteria. I will show that in a little bit more detail in just a second. So panel members can participate in actual judgments about the criteria in the evidence-to-decision framework, and you can choose which of those judgments you want to involve panel members at a given time. So there is guidance within GRADEpro. So if you, for instance, look at the interactive evidence-to-decision frameworks, this is from a screenshot from one of the recommendations. Here is a criterion. How substantial are the desirable effects? The panel may make a judgment about large, or you as a guideline developer leading the panel may suggest that it's a large effect, large benefit. So large benefit, and that means that you can then ask the panel about the agreement with this particular statement if you are the lead. So for instance, this is the detailed guidance for how to use it. You can list all of the criteria and ask panel members to either submit judgments about that criterion, or you can ask them for agreement with that particular criterion. So in this case, you would present no judgments to them, or you can actually provide judgments about certain criteria, or even about a recommendation. You can say, I suggest this is the recommendation, a conditional recommendation to use an intervention. And then you can ask a panel member to either vote or state agreement or disagreement with that. I won't go into the technical details. Once again, this is an update session, and this is well explained in the guidance of GRADEpro. So it might look like this. You will ask for either a judgment, this is what the panel member would see, or you would submit a judgment and ask them whether or not they agree or disagree with that particular judgment. So for instance, that the healthcare problem, is a priority. Okay. So with GRADEpro, you can also preview recommendations as you're sitting with the guideline panel, and then publish them as a mobile app. So for instance, at the very end, when you want to disseminate or preview, you can preview this by producing a QR code, take a picture of that with your phone, and look how it would potentially look. This is from an example from a guideline that we did with the Saudi Arabian Ministry of Health, where the recommendations are then produced as in an app. So for instance, here are different types of recommendations that a user of the recommendation could then use on the handheld or follow on the handheld, and you can preview this by using those QR codes and previewing before publication. So in this case, for instance, the panel recommendation is described here, the rationale and the benefits and harms related to that rationale. Okay, then finally, as part of an update for GRADEpro, and it now goes into talking about GRADEpro diagnosis and tests, I will use one example for how you can display information to guideline panels in order for them to better understand, for instance, information around test accuracy. I will use, as I said, one example. Interactive summary of findings tables are used to do that, and they are found under the comparison. This is where you produce your evidence profile or summary of findings table, as I said. So directly in GRADEpro, you can show information both about the effects of an intervention as well as some test results. I will move on to test results. So what we have found in quite extensive work with the GRADE diagnosis and healthcare-related test project group is that different users, different panel members need information to be presented, or want rather information to be presented in alternative ways. This is an example from using ultrasound for patients who are expected of having a deep venous thrombosis, and how you might present information to both your panel members to make a decision about a recommendation, as well as then for publication in an interactive format summary or findings table, as we call them. So we have found that users really want and need information presented in different ways. As a first example, we provide information as probabilities. So for instance, in a guideline panel, you may describe how a certain test result will influence the pre-test probability. So if your pre-test probability for the population that you assume to make a recommendation for is 5%, the properties of this test summarized in a systematic review would mean that the probability of really having a deep venous thrombosis with a positive test result is 68%. If you have a negative test result, the probability is still 1%. And the reason for that, as you know, is that no test or hardly any test is perfect, and that test results come with a post-test probability based on how likely an initial event is. We have found, as I said, that people want different presentations. So exactly the same data are showing here, and that is panel members and users of summary of findings tables might be interested in seeing how common is it that we make a correct diagnosis in people who have the disease or those who don't have a disease. Same numbers as we said before, pre-test probability 5%, same test properties applied. In 90.3% of people who do have a deep venous thrombosis, will you make the correct diagnosis? 10% will be either false positive, will be false negative in this case. And then those are the people without the disease in whom you're making the correct diagnosis, so some false positives, correct diagnosis. And then finally, our standard presentation of information, sensitivity, specificity of the test, and the results presented in frequencies. So different ways of showing the information. These are just 3 ways that are currently integrated in GRADEpro: probabilities, correct diagnosis, positive and negative test results, and you can go through a full evidence assessment in GRADEpro there, and then present the information in different formats, as I said, interactively. One word on a graphical representation. So for instance, if you chose the probabilities, you can then show your panel members that under certain or what that really means in terms of false positives and false negatives in order for the panel to make more informed judgments. Here's the overall quality rating. And then finally, plain language summaries. So we have provided a way, and this will be further updated in the future, to present information to panel members who are less numerically inclined and need narratives around it. That can be also done by actually clicking on the plain language summary and getting a result in narratives. Okay. This all then can be, once again, put into the evidence to decision framework and published on the database of evidence profile. I mentioned that we have a large number of evidence profiles in this great database. Your recommendation at the very end can be published and looked at by others, used by others, possibly modified. A recommendation might be looking at is depicted here, and you could then even look at the evidence to decision framework that accompanies it in order to understand the rationale for a particular guideline. I'll stop here briefly and just see what questions have developed. So I realized that in the previous question about the use of grade, few people had the correct answer. If there's time at the end, I will re-review this. The bottom line message from that was that grade can be used without systematic reviews, but we require we ask for a detailed description about the judgments that are involved and how the evidence was compiled. As I said, I will move on to an update about GRADE and non-randomized or observational studies. So a new tool has emerged. Many of you may have heard about it. A new tool that is not a great product, although a lot of people with great background have provided input in its development. And this tool is called the Robbins tool. You can find information on the following website, the riskofbias.info website with guidance. But essentially, this is a very interesting advance. It is a tool to assess the risk of bias in non-randomized or observational studies, so cohort studies and case control studies, for instance, interrupted time series. So, and the overall approach that these ingenious investigators have taken is that evaluations of the risk of bias, so of a detailed assessment of a study, can be compared to what would be the ideal study design. In other words, under which circumstances would available studies that are not randomized, so we are moving outside of the context of randomized controlled trials, how could they be compared against randomized trials, and how could we then assess how credible they are from the risk of bias domain point of view. So the evaluation of risk of bias in non-randomized studies are facilitated by considering each of these non-randomized studies, once again equivalent to observational studies, as an attempt to emulate or mimic a hypothetical randomized trial, so randomized experiment that compares the health effects of two or more interventions. And that tool is available, and GRADE had to really think about if guideline developers are starting, or systematic review authors are starting to use this tool, how can they actually integrate that with the GRADE approach? As in the risk of bias tool for randomized trials that people use, so for instance, looking at if concealment was done appropriately, randomization was done appropriately, risk of bias is assessed within specific bias domains. So once again, in the GRADE context, these would be risk of bias items. And they would be evaluated such that a hypothetical randomized trial is considered a target randomized trial. It may not exist. And a target randomized trial doesn't need to be feasible or ethical. It just is based on the foundational principle that randomization protects best against risk of bias. So in these risk of bias, replace this with items in the GRADE context. Within the risk of bias domain, individual items are assessed, and that would be done by outcome, so outcome 1, 2, and 3, across different studies, like we do in GRADE. So for instance, there may be 3 studies for which the risk of bias on these, based on these 7 items would be assessed, and that there may be different types of studies for another outcome. So this is mortality, this is stroke. We might include different studies that report on it. And finally, for the third outcome, yet again, a different body of evidence. These items, bias, for instance, due to confounding, selection of participants into the study, or measurement of the interventions would be assessed, and an overall assessment of the risk of bias would be done. Now, this has big implications for GRADE. The classic presentation of GRADE in terms of thinking about the GRADE criteria has always been, as you know, randomized controlled trials start as high certainty in the body of evidence for an outcome, observational studies as low certainty or low quality, and that was due to the fact that in these studies, observational studies, randomization was absent. And it was felt that that introduces risk of bias. But in reality, we then looked at risk of bias within these 2 bodies of evidence by, once again, considering detailed risk of bias criteria, that is the limitations in the detailed study design and execution. With a tool such as Robbins, this becomes quite differently. So the current approach has been For randomized controlled trials, we would use the risk of bias tool. For non-randomized studies, we would use a different risk of bias tool, such as the Newcastle-Ottawa scale or other types of tools. And then we would summarize this evidence and assess the risk of bias, here showing a Cochrane risk of bias table. And that would initially be done by study and then across studies, just like in the typical GRADE approach. Now, with Robbins, for those who do use Robbins, this is different, and we've described how it should be done. It is very clear, and we're doing this in one of the large projects, so further updates to come. It will require further development. That is, if you have assessed the risk of bias across studies with Robbins, so a different tool, then given that randomization is part of the assessment within Robbins, a different approach must be taken. And the advice that we currently give is that lack of randomization or allocation of the intervention is under the investigator control. It leads to a serious risk of bias or downgrading to low certainty by 2 levels, unless a rationale for not considering it is provided. And that needs to be made very, very clear. That is, if there is no downgrading by 2 levels, those who use Robbins should be justifying for not doing this. Because at the end, there would be an assessment of the randomized control trial based on the risk of bias tool, and then with Robbins or similar tools, if further tools will be developed in the future, all of the non-randomized studies would start as high certainty, but there would be likely downgrading because of serious risk of bias as a result of not randomizing. So this is how we describe it. No downgrading that is anticipated to be extremely rare. We have no examples. Even moderate risk of bias and downgrading by 1 level would have to be justified clearly, and we don't have any good examples yet. But this obviously, the use of Robbins, for those of you who use it, this has big impact on how GRADE is conceptualized. So the big question is, should this be done separately? Currently, if you don't use Robbins or a similar tool, there is no other tool available right now, this assessment would be done separately, and non-randomized studies start as high certainty. If you do use Robbins, one might have to combine these results, and under those circumstances, the proviso that I provided or the disclaimer applies. Non-randomized studies typically are lowered because of the various risk of bias items that relate to lack of randomization. But under those circumstances, if these tools are used, there may be a rationale for combining them in one assessment of risk of bias, both randomized as well as non-randomized studies. Okay, so I will come to my question. So once again, this is self-assessment for you on the call. I will go slowly. Another encouragement to submit questions or comments. The question period is interjected or is part of this actual presentation. So please do feel free to submit questions through the chat room or following the instructions that the event manager provided. So GRADE in non-randomized studies. The first statement, you will have to say which statement or statements are correct. Regardless of the risk of bias tool used, all evidence starts as high certainty. Second, if tools compare observational, so we call them non-randomized studies, to an RCT, which may be an ideal or hypothetical RCT currently in Robbins, then they start as high certainty. C, downgrading by 2 levels with the use of Robbins is default, and reasons should be described when it is not done. Which of those is correct, or is it B and C? I'll give you a chance to think about that and provide answers as well. I pause for 1 second. And I will now provide the answer. The answer also here happens to be B and C, that it is again B and C, as in the previous question is a result of randomness or chance, speaking about it. Okay, so I will move on to provide an update on what else GRADE is doing right now and what is new. I will speak about one of the GRADE project groups. That serves as an introduction. The GRADE working group with approximately 500 members who get together frequently just had one of its 2 or 3 annual meetings in Washington, which was supported by the National Toxicology Program with some travel awards. And one of the groups that presented at that meeting was the GRADE Animal Studies group. So GRADE has a number of groups and projects. Our new website is about to be launched, where you can get information about what groups and projects are part of it. And under the groups and projects that are described here include, for instance, our DECIDE research project that led to the evidence to decision frameworks. There are a number of other groups that are available and that GRADE members participate in. The ultimate goal is to provide GRADE guidance for these various topics. And as I said, one of them is the Animal Studies group. And I'll just give you a quick update on what these groups are actually doing. So the highly interactive meeting, GRADE for Animal Studies. This is based on a presentation by Miranda Langendam and Carlin Hoymans, who are leading this group. And although many of you are guideline developers, I thought it was really important to understand how evidence is assessed and moving a little bit outside of our realm of comfortness. So how can GRADE be used to, for instance, assess the certainty in the evidence from preclinical animal studies? And I keep this fairly brief, but animal studies in human health, including for guidelines, have quite a few roles. So for instance, drug research, experimental surgery, environmental health, so exposures are often, or harms are often evaluated on the basis of animal studies. But they can also inform clinical research in humans. And one of the areas, as I said, deals with that. So if you think of animal research, you can phrase questions in animal studies in exactly the same way as you would phrase the questions for human research, by specifying a population, an intervention, a comparator, and an outcome. As I said, particularly helpful for harms and toxic exposures. And when you think of that, the population, in this case animals, and GRADE has already referred to that in its paper on indirectness, can be indirect in many ways. So for instance, if you are looking at evidence from one species and compare it to another species, or if you're thinking about using animal research to move it into clinical decision-making, for instance, about clinical studies. So it can be used. GRADE is being assessed right now for use on when to move forward with human-based research based on animal studies. There are a number of challenges. This would be typical at a GRADE meeting. So for instance, how to identify all relevant aspects of translating, this type of research into human research. How to determine when differences, for instance, are substantial or more important than others, especially when you move across populations, or even when you move, as we said, from animals to humans. I'll give you a quick example. This has to do with the indirectness and inconsistency domain. Once again, many of these domains are related to each other. So let's just consider that you would have studies that would show the following results in different species. And you would be interested in moving research from animals to humans. So it's a translatability question. If you look at this evidence, this is a forest plot, obviously. Here are just the confidence intervals for the simplicity of presentation from, as I said, Dr. Langendam and Heumann. If you were to assess inconsistency and possibly indirectness, you would look at this body of evidence and you would quickly see, most people would agree, that there are different results by species. The question is whether or not you would lower your confidence. You would lower your confidence in moving forward to humans. And some of that might have to do with where you set your threshold. So are you just interested in seeing whether or not an intervention has an effect, and let's just assume that this is a positive effect if the effects are on this side of the line of no effect. And under those circumstances, you might say, despite the fact that the effects differ between species, there seems to be an effect and we might want to move forward. However, if you were to assess the effects within species, you would, just like on a population level, elderly, younger, children, adults, people with comorbidities, people without comorbidities, you might lower your confidence or your certainty based on inconsistency, which would be explained by the indirectness. In other words, you're generalizing from all species to humans. Here you would have concerns about, possibly concerns about indirectness, as opposed to possibly a situation like this where there is a big question whether or not to move forward because some animals, in some species there is an effect, in others there is no effect. Under those circumstances, between species you would definitely downgrade. Okay. I'll move on to the very final topic of the presentation today, and that is an update that is now, again, very relevant for very, very practical guideline developers. And that has to do with good practice statements or great good practice statements. And once again, just a reminder to submit questions through the chat room as they come up. A previous commentary published in 2005 was produced by some prominent GRADE members, which are listed here. It was not considered to be official GRADE guidance. But guideline developers out there have often struggled with when they should just make good practice statements, which I will explain in a little bit, and when they should go through the full effort of developing recommendations according to GRADE using evidence syntheses and assessment of the quality as well as evidence to decision framework. And given the interest, this was brought back to the GRADE working group, and an official statement by the GRADE working group, an updated statement is forthcoming. So here's the issue. What are good practice statements? The examples that we use in these presentations are as follows. So for instance, in endocrinology, you might formulate a good practice statement as follows. For patients with congenital adrenal hyperplasia, so that is an overproduction of adrenal hormones, we recommend monitoring patients for signs of glucocorticoid excess. Second example. Health services should be made available, accessible, and acceptable to sex workers based on the principles of avoidance of stigma, non-discrimination, and the right to health. Third example, in patients presenting with heart failure, initial assessment should be made of the patient's ability to perform routine or desired activities of daily living. One way of looking at good practice statements that Gordon Guyatt has used repeatedly is that if the alternative to the statement that you are making is laughable or not sensible, then that is a signaling question for making a good practice statement. So not monitoring patients for glucocorticoid excess, or not making health services available, or not doing a history and physical on patients with heart failure is probably laughable. What needs to be done in detail may be subject of a recommendation. So that can be characterized, as we said, when the statement would be informed by a wealth of indirect and linked evidence, and when there is really high certainty in the net benefit. In other words, when the benefit is clear or when there are minimal harms or costs. And finally, but a statement with caution, when it would be poor use of the guideline panel's time and effort to summarize this evidence. GRADE has now, as guidance, developed 5 questions that a guideline panel, before formulating a good clinical practice statement, should ask itself. And I will list these 5 statements. All of them should be answered with a yes. Is the statement, the good practice statement, clear and actionable? Is the message really necessary? Don't state the obvious. Not part of probably a guideline effort, unless, once again, it is really necessary. Is the net benefit large and unequivocal? Is collecting and summarizing the evidence a poor use of the panel's resources? That is, doing the review or collecting and summarizing, as opposed to referring, for instance, to indirect evidence that has already been synthesized or summarized. And then finally, is it possible to clearly and explicitly linking the rationale to that evidence, that indirect evidence that is being considered? So these are the 5 questions, and I will now, once again, for self-assessment, because this is a critical part of the GRADE approach, ask you once again one of the self-assessment questions. Good practice statements, GRADE good practice statements, are in order in the following situations. If the panels, for instance, don't feel like or don't have resources to review the evidence, or all of the criteria that I stated on the previous slide must be fulfilled, and avoiding systematic reviews is not the only rationale. I'll give you just 1 second to think about it. And I apologize for the simplicity of this question, but the main reason for this is we really want to make sure that good practice statements are not abused. They are necessary and important, otherwise we wouldn't be working on this topic, but they shouldn't be abused. So in other words, panels shouldn't make strong statements without I'm being very clear about it. So I need to ask the event manager. I just received a message that my audio was lost. Is the audio back, event manager?
It was just a couple of seconds, that's all. Thank you.
Couple of seconds, okay, okay. I read a couple of minutes and got very, very frightened. So can I just ask you again, was it on this slide or was it on the previous slide?
It was on this slide.
Okay, on this slide. Okay, very good. So once again, I'm asking the obvious question. This is because we feel it is extremely important that good practice statements are not abused. So the answer to this particular question is obviously B, all of the criteria that we described in the previous slide must be fulfilled, and avoiding systematic reviews is not the rationale. So I go back to these criteria for just 1 second. Statement clear and actionable, message really necessary, net benefit large and unequivocal, collecting and summarizing the direct evidence of poor use of panel's resources, and there is a clear link and explicit rationale of linking that indirect evidence or other evidence that is available. Will be described in an upcoming publication. The paper that I already showed is a good guide for now. And the answer was B, obviously. So I come to my summary. So I described new functions in GRADE. I described how the GRADE working group is working and what topics we are currently working on. Watch out for additional news. I described how GRADE may be used for rapid assessment and rapid guideline development. This is under development, and a current rough guide is available in Environmental Health International in a brief publication, but a GRADE project group will look at this in detail. When new risk of bias tools such as Robbins are used, observational studies start as high certainty in the evidence, but only then, and default is downgrading by 2 levels. It requires further guidance, but this is our current approach and guidance out there when you use Robbins. I talked about GRADEpro tests and interactive summary of findings tables and the importance of presenting information in different formats. I talked about GRADE project groups, so for instance, the animal research group, lots of work forthcoming, and I talked about good practice statements and an update from the latest meeting of the GRADE working group. We have some time for questions. I'm looking at the chat room and I'm not seeing any questions. Okay, there may not be any questions, and that is fine. I do hope that you are able to provide feedback on the webinar and possibly think about the self-assessment part that we have provided you with. This webinar was recorded, and you can go back and obviously look at what might be helpful in your future work. You can obviously also share this with colleagues. Thank you very much for listening. We just still wait for any chat questions that might be there. We have another minute or two. And if there are none, then we close the webinar, and I thank you very much for your participation.
Thank you, Holger. Ladies and gentlemen, that concludes your conference call for today. You may now disconnect. Thank you for joining. Have a very good afternoon.